And here we are with enzymes. So here's a protein diagrammed, and it looks like a complete nightmarish mess. It's got a very complicated shape, and we can describe the shape on 4 different structural levels. And by the way, I've just found out that I actually have a little pen that I can use, and I can scribble on diagrams and things. So, sorry, I just got okay, checking the chat right there. So in a model like this, we can describe this protein in terms of primary structure, which is the order that the amino acids are in, one after the other. And at this level, we're not even seeing individual amino acids. The chain of amino acids is just represented, by this long wire. That is one amino acid after another. Parts of that chain will kink up into things we call alpha helices. So that's an alpha helix, and that's an alpha helix. All of these things that look like phone cords are alpha helices, and we have a few areas like that one that fold up into beta sheets. That's actually supposed to be the Greek letter beta. And then things like that, we can represent with the Greek letter alpha, which looks like a little goldfish. And those are examples of secondary structure. And then the entire thing, not just local areas, but the whole thing will fold up into tertiary structure. And then several chains of amino acids, each with its own tertiary structure, may end up locking together, and we call that quaternary structure. It's a little bit like how you could describe a car if you needed to in terms of the nuts and bolts that make it up, or the parts, or the assemblies that make it up, like the drivetrain or the whole transmission, or the totality of the car. You could describe your car on multiple levels of structure, or you could describe, a building brick by brick, or, wall by wall, or room by room, or the entire floor plan. So you could look at a building on different levels of structure at once, and you can do something similar with proteins. There. And the point of all this crap is you make a ridiculously large number of different types of protein, and they do a ridiculously large number of things. They run your metabolism, they enable you to move, they help you regulate your body, they carry out homeostasis. Even deceptively simple bacteria might need thousands of proteins just to stay running. A heck of a lot of the secret of life is in the proteins. The function of a protein depends on its shape, and the shape of a protein depends on the order that you put the amino acids in. And what DNA actually does is carry the information that your cells can read and use to assemble amino acids in the right order to make a particular protein. And I want to spend the rest of the day looking at a class of proteins called enzymes. Goes a little like this. Oh, hi, cat. When you stick, sorry, I've got an annoying little white cat right here. Hey, kitty, come here. There we go. Good kitty. When you stick a bunch of atoms together, if you do it right, they will lock together and form stable bonds between them that we call chemical bonds. That's what holds atoms together in a molecule. We've talked about the very weak hydrogen bonds that hold the 2 strands of DNA together, and a much stronger, more permanent form of bond called a covalent bond. which holds together, things like the atoms that make up the sugars and the phosphates and things like that. For our purposes here, we don't need to go into what those bonds really are. They're usually drawn as looking like little sticks connecting little ping pong balls. If you want to know more about what chemical bonds are, we've got a perfectly good chemistry department over in Mannion Hall. You just need to know that atoms can stick to each other in a stable, mostly permanent fashion. And the thing about chemical bonds is that they represent energy, because when you break chemical bonds, you release energy. The simplest example I can think of is when you take very complex, large molecules of carbon, hydrogen, and oxygen, like the ones that might make up wood, for example, and you set that wood on fire. What you're doing is breaking the bonds that held together those carbons, hydrogens, and oxygens, and releasing a great deal of energy as you do it. And that energy takes the form of heat and light and motion, all these things that you see in flame. So energy is released when chemical bonds are broken. At the same time, you need energy in order to form chemical bonds in the first place. So when you do any kind of chemistry, you're basically breaking and releasing chemical bonds. Sometimes only breaking, sometimes only releasing, usually a combination of both. And there's a balance sheet when it comes to the energy that you're releasing. Just like a bank account, you might put in more than you're taking out, or you might take out more than you're putting in. The same is true for the total energy in a chemical reaction. If a chemical reaction produces more energy than it consumes, then once you start it, it's going to be like the Rolling Stones. Once you start it up, if you start it up, it'll never stop, never stop. Now, some of you are going to be earwormed with that. The reaction is self-propagating. Once you start a fire, the fire is going to keep burning until something puts it out or until it runs out of fuel. You don't have to keep adding energy to fire, because once fire gets going, it gives off more energy than it consumes. And a chemical reaction like that is said to be exothermic. On the other hand, a lot of chemical reactions consume more energy than they produce, and reactions like that need to have an energy source in order to proceed. We say that that reaction is endothermic. I'll be able to talk later. I'm leaving it aside for now, but your cells are able to carry out a lot of endothermic reactions because they're able to extract energy from an exothermic reaction called the breakdown of a compound called ATP. But we will talk later about ATP and what that actually does. And so we can draw this graph of the amount of potential energy in a chemical system over time. So we start with a certain number of chemical compounds over at the left. If the reaction is exothermic, then what we end with, the products, contain less energy, and so some energy is released, is given off during the progress of the chemical reaction. But we usually find that to get a chemical reaction started, we have to give it a little energy to get it going. You could think of this as, metaphorically, you could think of it as being like a ball sitting at the top of a hill. That ball will eventually, roll down until it gets all the way to the bottom, but you might need to kick it first in order to get it moving. You might actually have to nudge it over a little bump at the top of the hill before it can spontaneously roll down the rest of the way. So that bump at the top of the hill represents the energy that you have to feed into a chemical reaction to get it started before it can proceed spontaneously. And that energy that you have to feed it is called the activation energy. So, burning something, again, setting your neighbor's house on fire, not that I ever did that as far as anyone knows, is exothermic, because once you get it started, it will burn until something stops it or until there's nothing left to burn. However, houses do not spontaneously catch on fire because they need activation energy. You need to give that house a little boost of energy to get things started, which could be, a lighter and some gasoline soaked rags, or it could be faulty electrical wiring, whatever the case might be. You have to give it a little activation energy to get it going. When we have a substance that lowers a chemical reaction's activation energy, we call that a catalyst. To give you an example that you may know something about, if you drive a car that was made after 1975, which I assume almost everyone does, the engine has to be fitted with something called a catalytic converter. What that does is your exhaust passes through the catalytic converter, and inside the catalytic converter, there's a coating of metal. The best ones are use the metal platinum, although those are very expensive and some might use less expensive metals. The reason that's there is that your car exhaust contains one chemical is called carbon monoxide, and that's very poisonous. That's why you can die if you breathe car exhaust. There's also some other waste products in there, uncombusted hydrocarbons, never mind what those are, that just waste. And when you pass exhaust over your catalytic converter in the presence of oxygen, you can change those waste products into compounds that are less dangerous, notably a gas called carbon dioxide, which is still not really good for you, but it's much less toxic than carbon monoxide. So the platinum there acts as a catalyst because it lowers the activation energy that you need to convert carbon monoxide into carbon dioxide. And the catalyst is not affected by the chemical reaction. You don't need to get platinum added to your car's exhaust manifold. The platinum makes the reaction happen, but it's not burned up. It's not consumed by the chemical reaction. It's just there. And the reason all of this matters is that enzymes are proteins that act as catalysts in living systems. Enzymes are not used up, although you do have to periodically replace yours, but they're not used up in chemistry, but they make chemical reactions happen by lowering the activation energy that you need to get the chemical reaction started. Incidentally, there are also some small molecules that are not proteins, but can play an enzyme-like role in making chemical reactions happen. We call those coenzymes. And the only reason I mention coenzymes is that a number of vitamins that you might know happen to be coenzymes. Your B vitamins, for example, are not proteins, but they can act like enzymes, and we call them coenzymes. So ultimately, that's why you need B vitamins, and I think some others as well. Right. So if there's no activation, if there's no enzyme present, this chemical reaction can't really get going because it has to get all the way up and over this hump in order to proceed. It's like a ball at the top of a hill, but it can't get over this little hump at the top of the hill. So it's kind of stuck right here. Here's our little soccer ball. Sorry, trying to make it look like a soccer ball. Close enough. There we go. there's our soccer ball. It's at the top of the hill, but it has to get up over this hump before it can roll all the way down. And if nobody comes around and kicks it over the hump, it might be waiting there an awfully long time. What an enzyme will do is get rid of that hump and lower the activation energy, maybe even lower than that, make it so low that it's much more likely that that ball is going to get over the hump and roll downhill. And in chemical terms, that makes it much more likely that the chemical reaction will proceed. So the chemistry of your life mostly happens because you make enzymes that make it happen. Okay. Enzymes can't act on just anything. They act on very specific molecules that we call the substrate of the enzyme. And the way we think these works, this is why shape is so important. Enzyme molecules can have a bunch of crazy shapes, but there will be a spot on an enzyme molecule called the active site, and the substrate fits into the active site like a key fitting into a lock. Sorry, if the slides are shifting at odd moments, it's because I've got a cat here that is poking at me and has decided my hands are delicious. Anyway, so the substrate will fit into the active site on the enzyme, and once it's done that, the enzyme molecule might put stress on the substrate and cause it to break at a particular spot. Or the enzyme might take the substrate and move it close to another molecule and make those two stick together. So enzymes can build up as well as break down. And then once that happened, the substrate falls out of the active site. It may have either been broken down or it may have been put together, as the case may be. Enzymes can do both. And here's a diagram of how this looks. In gray, you've got an enzyme molecule, and it's got this funky shape. And then the substrate fits into the active site. The enzyme may actually change its own shape just a little bit as the substrate binds in there. We call that induced fit. Don't worry about that here. And once the substrate is in the active site, the enzyme may encourage the substrate to break, which is what's shown here. And that substrate breaks apart, and then the two broken pieces fall out of the active site of the enzyme and go on their merry way. Alternately, you could have two different substrates fitting into the active site, and the enzyme sticks them together. That could also happen. And you could compare enzymes to being like little socket wrenches. The wrench itself might have a number of shapes. The handle could be different thicknesses or different sizes. But what's critical is the socket on the wrench. That would be the active site because it's able to fit only one size and shape of nut or bolt or what have you. The nut or the bolt would be equivalent to the substrate. The nut and the bolt fits into the active site, and then the wrench can be used to turn the nut or bolt or whatever it's made for. So there's a decent concrete model for how enzymes can be thought of as working. It's not an absolutely perfect model, but it will do for our purposes here. Okay, here's an actual one, and I picked this one because it's very familiar to you because it's in your saliva. You start digesting your food the instant you put it in your mouth. Saliva contains enzymes, and what they do is break down the large molecules in your food and turn them into small molecules that your body can absorb and use for its own purposes. So you've got alpha amylase in your mouth, even as we speak. And you can actually feel a little bit of how it works if you put a very starchy food, like a saltine cracker, into your mouth and you start chewing it. If your mouth is dry, the cracker is not going to taste like anything. Once the cracker starts getting moistened with your saliva, countless millions of molecules of alpha amylase start latching on to molecules of starches and complex sugars. In this picture, the alpha amylase is shown in blue. The active site is a groove right there, and fitting into the active site, that thing shown in yellow, that is a short molecule of starch. So the starch molecules fit into the active site, and the alpha amylase begins to break those starches down. Starches are polymers of sugars. So the alpha amylase starts chopping up that starch, breaking it up into sugars, and you can actually taste this. If you pay close attention to what it tastes like to eat a salted cracker, at first it doesn't have much taste, but you start getting, aside from the salt on it, you'll start getting a bit more flavor, maybe even a slightly sweet taste as you chew that cracker and it gets moistened by your saliva and turns into the pulp that you end up swallowing. That effect is due to the alpha amylase in your saliva. And now you know. Here's another one that's important. This is an enzyme that certain bacteria make. It's called beta-lactamase. Here, the chain of amino acids is shown in blue. The parts of the chain that have kinked up into alpha helices are shown in green. And the parts of the chain that have zigzagged into pleated sheets are shown in red. And if you look over on the left-hand side, sorry, hang on, I'm having a little trouble pulling up my trusty pen. Okay, I'm going to start this again. Here we go. Okay, so things like in green right there, there's a nice alpha helix for you. All these red things over here, those are beta sheets. And then this little stick-figured thing that looks mostly yellow, that's a molecule of an antibiotic. It's called penicillin. And the penicillin fits into the active site, which is right here. This thing that I'm tracing right there, that is the active site. That's where the penicillin fits in. And when the penicillin fits in there, beta-lactamase strains the molecule and causes it to break. The reason this matters is bacteria that can make beta-lactamase can break down penicillin instead of getting killed by it. In fact, there are some bacteria that can eat penicillin. This is one of a number of the tricks that antibiotic-resistant bacteria have. This is one reason why it's not that common to treat infections with ordinary penicillin anymore. We've had to develop new antibiotics because more and more bacteria are showing up with the ability to make beta-lactamase. So if you look at all of the metabolism that's going on in a cell, you could think of it as being like an enormous set of assembly lines. Here's a bunch of, looks like cars being put together. And every one of these yellow things is an industrial robot. And each one of those does exactly one step. So a car moves up to one of these robots, and the robot will stick out an arm and weld, zip, 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 zip. And then the car moves on to the next robot, which does something different. So on this assembly line, every industrial robot does one thing, and then the car moves on to the next one until it's assembled. In every cell, every enzyme would be a robot that carries out exactly one step of metabolism. And the totality of a cell's metabolism looks like this. And I hope you're copying all of that down. Actually, I'm kidding. You don't need to copy all of that down. That thing looks like a horrible mash of spaghetti, and it's hard to see detail at this level. But every one of those thin lines that you're seeing is an arrow representing a chemical reaction, starting with one compound and changing it into something else. Every one of these little lines represents a chemical reaction. Every one of those reactions happens because an enzyme catalyzes it, makes it happen. So that you can see right away that just to carry out the most basic metabolism, a cell has to make, I'm guessing, hundreds, if not thousands, of different enzymes just to break down its food, extract energy from it, turn whatever it's feeding on into substances that it can use and build up molecules of its own. All of that catabolism and anabolism relies on thousands of enzymes, each of which does one thing. So that's your assembly line for life right there. All right. I'm going to take some of this and turn it into a familiar, a context that I think many of you will be more familiar with. Let me see here. If we go back to this thing right here, let me see. Somewhere, I think it may be this. If you were to take those chemical reactions and zoom in on them, they would look like this. By the gods, don't try to memorize these names or these steps. The most basic fuel that typical cells use is a simple sugar called glucose. Glucose doesn't taste quite as sweet as table sugar, but you can certainly eat it. If your cells are not able to take up glucose, it ends up in your blood and your urine, and you have diabetes. So you might have heard of that. And glucose gets broken down into two molecules of a compound called pyruvate. All right, just checking the chat to make sure everything's okay. And it's broken down to pyruvate in 1, 2, 3, 4, 5, 6, 7, 8, 9 steps. Every one of those steps is represented by a red arrow. So glucose gets converted to glucose 6-phosphate. Glucose 6-phosphate gets turned into fructose 6-phosphate. Fructose 6-phosphate gets turned into fructose 1,6-bisphosphate. That gets broken into two molecules of, I've just forgotten what GLAP stands for, doesn't matter, on and on until you end up with pyruvate. The whole thing, all of these 9 steps together, this one little part of the assembly line is called glycolysis. And every one of those steps happens because there's an enzyme to make it happen. The enzyme names are shown in red here. This ultimately results in generating chemical energy from the food that you eat. You couldn't live if your cells could not do this. I'll go into exactly what I mean by getting chemical energy out of the food you eat. Let's just say this is part of what your body does in order to fuel your metabolism. Sugars that you eat get turned into glucose, and the glucose gets broken down into a molecule called pyruvate. There happens to be a species of yeast. If you remember, yeasts are single-celled fungi. This one is called Saccharomyces cerevisiae. The cells are very small. They're something like 5 microns. That's millionths of a meter. They reproduce by forming little buds, which is what they're doing here. And the reason why Saccharomyces cerevisiae is so important is that if you feed it sugar, it will break that sugar down into pyruvate by glycolysis, the same way that your own cells do. But it will follow up glycolysis with another set of reactions called fermentation. And so yeast will take that glucose and turn it into pyruvate, and then ferment the pyruvate, goes through several steps, and turn it into carbon dioxide gas and ethyl alcohol. Ethyl alcohol being the stuff that people have been known to drink. So if you take a bunch of Saccharomyces cerevisiae and turn them loose on a liquid that is rich in sugar, grape juice, for example, and let them do their thing, the yeast will break down the sugar in the juice, break it down into pyruvate, and then ferment that pyruvate into ethyl alcohol, and lo and behold, you've got wine. If it's a still wine, they ferment it into ethyl alcohol and carbon dioxide gas. If you let the carbon dioxide escape, you've got a still wine. If you trap the carbon dioxide gas using a special bottling method, you've got champagne, or asti spumante, or other forms of sparkling wine. If you do this with other fruit juices, you have fruit wine, blackberry wine, or strawberry wine. Sorry, old song. If you do this, turning your yeast loose on honey and water together, you get a drink called mead, one of my favorites. This is a grain, Hordeum vulgare, otherwise known as barley. Barley does not have a great deal of sugar in it, but what it does have is lots of starch, and starch is a polymer of simple sugars. Take a bunch of sugar molecules and string them together, and you end up with starch, and there's lots of starch in barley. That sugar is what the barley is going to use as fuel when the seeds sprout, right? A seed has to have an energy source until it can put up leaves and start doing photosynthesis on its own. So every seed contains a fuel supply. That's what the starch is doing in barley. So if you take barley and you soak it in water, and then you spread it out like this guy is doing, and you let it dry, the seeds will begin the sprouting process as soon as you soak them in water. And you don't want to let this happen for too long. You don't want your barley to actually start growing leaves, but you do want your barley to begin the sprouting process, and it starts by producing enzymes to break down that starch. That starch has to be broken down into sugars before the barley seed can use it to start growing. So the first thing each seed does is start cranking out enzymes. And by the way, when barley has just begun this process, we call it malt. So this is barley grains in the middle of the malting process. And once barley has started malting, the seeds begin to produce enzymes called amylase. That's the same word, although not exactly the same shape as the amylase enzymes in your saliva. We're not going to use saliva for this. But the amylase enzymes that are produced by the seed, which are shown here in blue, latch on to molecules of starch and start chopping it up into a simple sugar called maltose, which the barley seed can then use as an energy source to grow its first roots and shoots. By the way, I don't know how many of you have done this, but you can make syrup out of barley grains by malting the barley and then kind of grinding it up, and you make something called malt syrup. I don't know how many of you have done this, but if you've ever gone to an old time ice cream parlor, they might make you a shake or a malt A malt is an ice cream concoction that has some malted barley syrup added to it for extra sweetness and flavor. So if you've ever, I don't know, watched a movie about the 50s and, the guy and his girl walk down to the malt shop for an ice cream malted milk, that's what they're getting. They're getting like a milkshake made with some barley syrup, where the amylase has been allowed to break down that starch into maltose, which is sweet and flavorful. Now you know. This is the other thing that malted barley is good for. You grind up malted barley, you soak it in hot water to create a sweet extract that's got lots of maltose in it, and then you feed that sweet extract to Saccharomyces, the yeast, and the result is beer. You being college students, I'm sure you've heard of beer. Now, this being a dry county, I'm sure you're all law-abiding citizens who would never think of touching a glass of beer until you are of legal drinking age. But I'm assuming you've heard of beer at some point. And this is how beer is made. You make living barley seeds crank out enzymes. They chop up that starch into sugars. You feed the sugars to yeast, and what you end up with is alcohol and carbon dioxide from fermentation. Mass-produced beer is made extra fizzy because they pump carbon dioxide into it. But if you've ever had bottle fermented or cask conditioned ale, they call it, that just retains the carbon dioxide from the fermentation process. So that's how you make beer. Stronger drinks are made by brewing a beer or a wine and then heating it to concentrate the alcohol and get rid of the water that the alcohol is blended with. So you basically brew a beer and heat it and condense the vapors. We call that distilling it to make whiskey. If you do that with wine, you get a brandy. And both whiskeys and brandies may be stored in a wooden barrel for some time because that improves the flavor. So yeah, here's some locally made whiskey, by the way, from the only distillery in Little Rock, Arkansas. Not that I'm encouraging anybody to try this because, you all have to be of legal drinking age, but I'm just saying this is, one example of this that we make around here. Okay, right. All right, I tell you what, that's actually a pretty good stopping place. I haven't said everything I really wanted to about enzymes. I kind of got carried away talking about the joys of alcoholic fermentation. But I'm going to go ahead and stop there because it's almost 10:50. We will pick up the rest on Monday. Don't forget the exam is this Friday. And by the power vested in me by the state of Arkansas, I will here stop the recording.